Alright, so in today's video, we're going to look at gram-positive and gram-negative cells. Throughout the video, we're going to learn about differential stains, the difference in gram-negative and gram-positive anatomical structures, and their laboratory use. So gram-positive cell. The first thing we need to know about it is what is a differential stain. So a differential stain is a staining process that utilizes more than one chemical stain. By using multiple stains, it becomes possible to differentiate between different microorganisms and or their structure. So the gram-positive cell, when you go through the staining process, it's going to stain purple. Now how do we remember this? So what I like to do is associate purple with the color of royalty. And old times, royalty wore purple. So when you think about that, it'll be a good way for you to remember that purple equals gram-positive. Now, the next thing we have is a gram-negative stain. The staining process is the same, but with a gram-negative cell, it's going to stain pink. And this is due to some anatomical differences. So, within a gram-negative cell, it has two membranes and a thin peptidoglycan layer. Now the picture we see here is what you might see under the microscope. So we have some different morphologies and different shapes, different clusters, things like that. So what we see here is a pinkish reddish color, and this is how you know that it's a gram-negative cell. And what I like to do is associate this with a blister, kind of like you got burnt. So when you think of gram-negative, think of that pinkish colored blister. So the next thing we have is a gram-positive cell and its anatomical structure. So the main thing here to realize is there's a thick peptidoglycan layer, around 60 layers, and this causes some of the staining difference. So other things we have are lipoteichoic acid, teichoic acid, membrane proteins, your phospholipids. Your cytoplasmic membrane is a lot thinner, and there's only one in a gram-positive. And then you have things like your porin that allows for the free flow of molecules and different things like that. The thing we have here is the gram-negative anatomical structure. So the picture we have here is a drawing of the cross-section for a gram-negative cell. So, like gram-positive, you have your cytoplasmic membrane, but as you move up into their periplasmic space, where the peptidoglycan is located, you only have around 3 layers, so it's much thinner than gram-positive. And within this periplasmic place, where peptidoglycan is located, this is your assembly and disassembly location for some of your cells, or different molecules and things like that. So as you go up, you have things like your porin. Those allow, again, for the free flow of molecules in and out of the cell. But when they get into that periplasmic space, those enzymes and things like that are going to either break down or allow those things to go to the different cytoplasmic membrane. So the next thing we have is our LPS. So with our LPS, we have something called lipid A. And lipid A is actually an endotoxin, and it's released when the cells die. So when you have a bacterial infection that is gram-negative, doctors really have to be careful about how they treat that, because if they kill the cells with an antibiotic, then they're releasing that endotoxin, and it can cause fever response, shock, and different things like that. Then we move on to our core polysaccharides, and this is just repeating subunits. And then on top of that, we have our O antigen. And what that is it's kind of like wardrobe change. If you ever see behind the scenes of theater or concert or anything like that, you're going through constant changes in what you're wearing. So they will go through that process, and that kind of helps protect the cell, because then the antibodies can't really distinguish what they're looking at. So how do we use this in the lab? So there are certain steps you're going to go through to perform a gram stain. The first thing you're going to do is draw a circle on your slide with a Sharpie. And that's just kind of giving you reference for where you placed your bacteria. The next thing you want to do is collect a small drop of water with your loop. Now your loop is just a laboratory device. It's a handle with a thin piece of wire with a little circle on the end. You don't want to collect too big of a drop of water, and when you place it on your slide, make sure that spread out is about as even as you can get it. Now, this is only if you're working from bacteria from a petri dish or a plate. So the next thing you're going to do is aseptically collect your bacteria. So what I mean by aseptically collecting your bacteria, you're going to flame your loop with your Bunsen burner. You want to do this at a 45-degree angle, and you're going to flame from base to tip, making sure you get it red hot. And that heat's going to kill any potential contamination. And then you're going to let that cool a little bit because you don't want to place that straight into the bacteria and let that get airborne. After you've collected your bacteria, you're going to smear that in with your water on your slide and let that completely air dry. Once it's air dried, you can then heat fix it. So when you heat fix it, you're going to get your slide holder, or it could be a clothespin. So when you grab your slide, you're going to pass it through the flame around 2 or 3 times just to get that bacteria really set into the slide. Now the next thing we have to do is prepare our stain. The first thing you're going to do when you're preparing a gram stain is you're going to locate your crystal violet stain, and you're going to place that on your slide where the bacteria was located, kind of flood it, and then you're going to let that sit for about 1 minute. Once it's set, you're going to rinse it with water, making sure you get all the stain that you can off. Then you're going to move on to iodine, and you're going to set that in the bacteria as well. Once you have set that on there, let it set for about 1 minute, and then you're going to rinse that with water as well. Next thing you want to do is decolorize with acetone alcohol. Now, personally, I like to do about 10 seconds. You know, it says 8 to 15, 6 seconds. 10 seconds is about good. Then you're going to rinse with water. Now, this is actually going to wash your Sharpie off, so you really need to know where you placed your bacteria. Next thing you're going to do is you're going to stain with safranin for about 1 to 2 minutes. About 1.5 minutes is sufficient. Then you're going to rinse it with water as well. After you've completed this process, you're going to blot it dry. You want to get your slide completely dry. That way you can view it under your microscope and the water isn't causing any refractive indexes to lose the image. When you're viewing it through your microscope, you're going to have to get to your oil immersion lens to be able to clearly see if you're working with a gram-positive or gram-negative cell. Now, the key to this is to go from your lower objective lens for about 10x and then follow your way up, making sure that each time you move up, it is focused and centered on what you want it to be. And then on the right here, we have a picture of what could possibly be your gram-negative or gram-positive cell. Here we're looking at gram-positive. You can see all the purple coloration. You should also be able to tell the morphology of the cell, whether it's rods, cocci, things like that, and as well if it's clustered. So if it's streptococcus, staphylococcus, and different things of that nature, you should at least be able to tell this about your cell. And then if those are pretty much the introductions to what a gram-negative and gram-positive cell are. As we go through the semester, we'll use these throughout the laboratory techniques. If you have any questions, please let me know.